be the chair of this session. My name is SANDRA LEJNIECE. I'm professor and dean in Riga Stradiņš University, and I also lead chemotherapy and hematology clinic in Riga East University Hospital. We will have 4 speakers for this session, and all questions will be later after all talks. We will hold discussions. The first speaker will be Professor David Gisselsson North from Sweden. He is the Vice Dean Principal Investigator in Lund University Cancer Center, and he is also interested in all children diagnosed with cancer in Sweden genome sequencing. Please, the podium is yours.
Thank you very much. It's a great honor to be here. And I will today brief you about our project, a national project, where we have implemented whole genome sequencing for all children with cancer in Sweden. I happen to have the pleasure of being the coordinator of this project. There's more than 100 people involved in Sweden. And one of the other major players and coordinators of this project is Johanna Sandgren, who is here today in the audience. You can ask her as well about this project. So this project, which was set up and really started in 2021, had several aims. One was really to try to set up whole genome sequencing and also RNA-seq of tumor RNA to increase the diagnostic precision and risk stratification of children with cancer. So this makes it very different from many of the other similar projects that had been launched before, like in the US or in Germany, where you had INFORM, where high-risk patients were selected, typically actually at relapse, to have their genomes and the tumor's genome sequenced. So what we said, we start up front when the kid is first diagnosed. On the first biopsy, we do the sequencing to see if it helps us also in the diagnostic process. And this makes this project very different from other published data sets out there. Of course, we also wanted to detect targets for precision medicine like the other studies, and we also looked at the germline sequence, looked for relevant hereditary conditions, and also in this project, we collected all the DNA sequences and RNA sequences at a national resource in the pediatric solid tumor biobank in Solna. And of course, we also wanted to evaluate the future potential value of large-scale sequencing in general. So in Sweden, we got extra government funding to do this in children as a pilot to see how we could use whole genome sequencing in the adult setting. Now, some challenges here is that Sweden is a dispersed region with a regionalized healthcare system. So we have 6 pediatric oncology centers in Sweden. You had to make them work together. Sequencing is now being done in 4 places, but all centers are really participating. That's one of the challenges, and we have no really top-down governmental steering of healthcare. It's all regionalized in Sweden, so you have to talk to all the healthcare regions on their own. We had some logistic issues, of course, to send to sequencing sites, but that was kind of easily solved. Something that was a bit more problematic was to have the head of departments out there in the healthcare, the ones responsible for the budget, to kind of embrace this project. Because even though it was funded from the government and from the Swedish Childhood Cancer Fund, a lot of hospital staff had to work on the project on their hospital time. And then, of course, their superiors had to agree to do that. And we also always got this question, is this a research project or is it clinical? And we said, well, we're implementing this in the clinic, even though it is also a research project, because the evidence is there that it will probably be very clinically useful. So this was really a gray zone project, and healthcare officials really has problems with the gray zone. Methods were expensive, but we were very well funded to start with, still are. And methodology expertise, we were worried that we wouldn't have enough bioinformaticians, for example, but that was kind of largely solved. Actually, Sweden is a great country to work in. We are good at recruiting people. And we did have skepticism, especially among oncologists, about the issues that Professor Green actually talked about before, saying, there will be like a flood of data coming over the oncologist, and we will have all these genetic diseases that we have to handle. But well, the skepticism has actually completely now changed into big enthusiasm among the oncologists who are really now, so I'm a pathologist, I kind of include patients in the south of Sweden, and they call me up and they want this sequencing done, even on patients where it's that where there's really no indication, maybe they have a benign tumor, and they still want it done. So they have been almost addicted to these new methods. The setup is kind of very, very regular. You take a tumor sample. This is for solid tumors. We also do leukemias now, but for solid tumors, we have to first check that there is enough tumor cells in the sample. So here it's really the pathologists. So there are like three pathology centers that really look at the slides here and say, oh, is it stop or go? Is it more than 40% tumor cells, and is it malignant? And then we include it. In one of the centers, we even now include patients even before the informed consent is being signed, saying, okay, this is really healthcare, and if they don't sign informed consent, they will not be included in the research part, not in the data sharing, but the rest can be done as healthcare analysis, and the hospital will take the costs. And that actually reduces our turnaround times locally to quite an extent. So it has to be malignant, more than 40% tumor cells, and then we run whole genome sequencing, RNA-seq, and DNA methylation analysis if it's a CNS tumor. Compile this together in a report at the Molecular Tumor Board, which is held online. So this afternoon I will be away for an hour to have our Molecular Tumor Board online with pseudonymized data, which is very practical. And then we sign out a report, and it reaches the oncologists, and then we have a few patients that have to go to genetic counseling after that. So some data after the pilot year. So 2021 was more or less a pilot year, and after that we summarized our data, and this has been published already. And something that really made us happy when we looked at the first 118 solid malignancies from 117 patients was that we had an equal inclusion rate from the different centers. We talked about this before, that maybe you can set something up in a cool tertiary hospital where you really have like a comprehensive cancer center and its clientele of patients, but what about an entire country? If you can you go out in the countryside? And yes, we can. There's no difference between the different sites. So even the small countryside clinics, so to speak, are also included as much as the university hospitals here, the big university hospitals. We had a good distribution among the different pediatric cancer diagnosis, except for lymphomas, where the tumor cell content on a regular basis is quite low. You know, Hodgkin's lymphomas, for example, just 1 to 2% tumor cells, we can't include them. Otherwise, we have all the entities there, sarcomas, rare carcinomas, anything, and that was really good. We found something of clinical significance in the somatic tumor genome when filtered against the blood in actually 91% of included cases that either corroborated the diagnosis that were already set histologically, or that added something new that we didn't really know. And we found potential drug targets in 24% of included cases, but far from all these patients actually got precision drugs, because many of them did well on the ordinary therapy. And then we had some notable surprises in between the expected targets, and I will show you that. But something that was very interesting from the perspective that this was a unique study where we did this really upfront, including all patients, not just the relapses, not just high-risk patients, was that in 60 of these 113 informative patients, we could actually get additional diagnostic information from risk stratification that helped the diagnosis. So this is really helpful to pathologists. And this was not shown before, really, that you can really get a lot of value if you do this upfront from the beginning. So that's helped us out. This was our first endpoint. So where are we now in this project? That was the pilot year. We went up to full speed. We covered around 80% of patients in Sweden. The rest had too low tumor cell content with childhood malignancies. Starting from February 2022, then we started to getting that high coverage. We kind of reached the technical ceiling, really, and I think I only know one patient where the family opted out and did not participate in the study. But when with the last summary, that was now in spring 2023, we had sequenced more than 300 solid tumors and 100 leukemias, more than 300 germline genomes analyzed, and then inclusion rate had reached what we now have the technical ceiling of around 80%. We actually had more patients now. In the first study, we only had one patient, but now we have 7 patients, actually, where the morphological diagnosis was revised to something completely different based on sequencing. So we thought from the beginning that, we are kind of good pathologists, just one single case with revised diagnosis. That's good, but now we have more. So yeah, it really helps. And these are experienced pathologists, that look at these samples. 22 patients had therapy modified because of these findings. Most of them got targeted drugs, I will show you, but we also have other modifications. We had clinically relevant germline variants that had not been noticed, that were not known before the kid got cancer in 8.5%. And the data is being uploaded as planned to the biobank. And I will mention something about the costs and the turnaround time, which are practicalities here. And to start with, the turnaround time is kind of approaching 3 weeks in all of Sweden as a median. And the costs is actually not that high. So for Sweden, we think that if we do all the solid tumor, it will be a bit more than €1 million with current sequencing costs, including personnel costs and storage and so on. So it's really not that expensive, maybe around €2 million when we go to covering also leukemias. This is the turnaround. This is the pilot year, right? And then we go down, and then we'll be nudging down to 3 weeks on average. But actually, in small centers, we are now down to 8 days of turnaround time, which makes this very, very useful. So it comes back around the same time as the latest immunostains in pathology, which is kind of makes this fairly practical. Some examples of precision treatments that we find. So these are the CNS tumors. It's, of course, our major group with the solid tumors, where you see that you have all the white ones, or we find a somatic mutation that's an indication for a targeted drug, and we have plenty of variants of those. Something which is, many of those are like the expected, like NTRK fusions and larotrectinib, but then we also have the green ones, where actually we treat on indication where also the germline genome contributes, and that's so, especially we find the microsatellite instability syndrome, so they can get immunotherapy, which is fantastic. We have quite a few of those patients. And then we have the yellow ones, which with the therapy, not targeted drugs, but the therapy is modified because we find the germline variants. And they are actually kind of a common groups. Got the neuroblastomas with ALK mutations, that's kind of really expected here. Sarcomas, kind of a few rare things. Targeted therapies, and then lymphomas, and then other entities like rare carcinomas. So these are the 22 patients we know of. I guess we are now in autumn, so it's maybe 30 patients with special treatment. These are just some examples of surprises that we found. This was our first patients with a revised diagnosis with a really tricky spindle cell morphology. Patient with a tumor in the neck, young patient. And we were kind of really convinced that this was an infantile fibrosarcoma, and the immunostaining really supported that. But the whole genome sequencing found a really strange X18 rearrangement with actually an SS18 fusion. So this was a synovial sarcoma, it turned out, with a completely different treatment. So this patient would not, I think, have survived if we set that patient on the infantile fibrosarcoma treatment. This is another example where you find something really, really odd. So this was a tumor that first looked like a rhabdomyosarcoma, but the immunostains didn't really fit, and we found this really rare fusion gene, EWSR1 KLF15. And with that one, it was actually saying that, oh, this is a specific type of tumor that is signified by this fusion gene. And how to treat that one, we didn't really know, because this girl had really progressive disease, regular chemo didn't help. And then we found a single case in the literature, I think from the US, where a patient with this one had happened to be enrolled in a trial with pazopanib, pan-TRK inhibitor, and did really well. So we used this drug just on a clinical trial consisting of one patient, and it actually worked. I mean, she's still alive. So it's kind of, yeah, this was maybe incidental, not really evidence medicine, but we're at the forefront, so these things happen. So the take-home message from this is really that it helps. If you do it up front, include everyone, you get a lot of help in the diagnostic process, even in this unselected cohort of just childhood malignancies overall. We are right now in discussion with the local politicians about whether this should be in the standard healthcare budget. We have quite high hopes that this will be solved, at least with 1 or 2 years from now. But we also see that our clinician must really learn to leverage this new genetic data, because we see a difference between the different places, the different hospitals, in the sense that in some of the bigger hospitals, the clinicians are really, really keen to use targeted new drugs, but in the smaller hospital, not so keen. So we start to see differences there. So we have put a lot of efforts into educating our oncologists, but we have to do this even further, I think. So what's happening next if this becomes standard of care in Sweden, which I think it will pretty soon, with hospital funding? And what we're doing now is to saying, okay, so we still have a lot of challenges in pediatric oncology, so maybe we should think about integrating data modality, not just looking at the genome. We have some really big challenges in pediatric oncology. For example, how should we phase in precision drugs and replace the old therapies earlier detection, especially of relapses, could make a big difference. How should we do that? How should we dynamically change treatment as a tumor evolves? How should we introduce evolution-based treatment? And how should we kind of have novel innovations, technical innovations in the field? We said for a small country like Sweden, even with great collaborators, we are right now collaborating with Genomics England, for example, and France Genomique, still, I mean, what we can do is to add more modalities to our data. We don't have that many patients. And for that, we are trying to sketch out something called the Brainchild Metadata Catalog project by the Swedish Childhood Cancer Fund, which support this, also in collaboration with the Swedish big initiative called the Data-Driven Life Science Initiative, with a lot of private investor money, and the SciLife Labs, and really try to get data together from pathology, radiology, sequencing, and other data sets. I mean, you can see in the future there will be wearable devices and so on. So this is just a sketch right now, and I would love to have people to come up to talk to me about this, because we really think that if we could create a really good multidimensional data set, even for maybe just 1,000 patients, this is Sweden, overall a small country, we could still have a very unique and interesting data set for training machine learning applications. So that's for the future. We will start working with a metadata catalog in this next year. So that's something, that's our vision right now, to kind of shift, putting genomics aside, making it standard of care, and then go into machine learning, the new world with multidimensional data. Yeah, that's it for me. More than 100 people were involved. I just happened to be the spokesperson. And please talk also to Johanna Sandgren about this, who is here today, and also the SciLife people who will arrive later today, Päivi. Thank you.
Thank you. Thank you. And next speaker will be Martin Zoche. He is Director of Molecular Tumor Profiling Department of Pathology and Molecular Pathology, University Hospital in Zurich, Switzerland, and also lecturer at University of Zurich and postgraduate study course in Pharmaceutical Medicine University in Germany. Please.
Thank you very much for this very kind invitation. I'm also the second time here in this forum. I remember very well last year, October, and I think the weather was a little bit better last year. But everything else, again, is really, really fine. I had a lot of very nice discussions. Today, I want to give you a new overview about our newest project. We are merging now clinical data for oncology patients. And I want to show you what this means and how we cope with these huge amount of data and sort and organize these data into databases so that these data are for later research studies comparable, usable, and research can be done on the data. Just to give you an overview about how many and how much data we are really scoping about. So we had a tumor profiler project, TUPRO, study in our university hospital, and more than 500 patients were diagnosed on a very, very deep level. I do not want to go into all these details, but it was done much, much more of diagnostics than just the routine diagnostics plus genomic sequencing. So just to give you an impression, the diagnostic for one of these patients costs more than 100,000 Swiss francs we have spent to diagnose the patients in a very, very deep way. And you can imagine that doing all these different kind of tests, a huge amount of data are created. So you can see it down here. For each patient, we have created more than 500 gigabytes of data. And when you gather all this information together, and in the end, you have the molecular summary report, the tumor board report, which is only 1 megabyte long. It's a PDF file. And you can imagine that you have to follow down and to condense all this data is a huge effort without losing important information. I think this is a very important part because you do not want to lose the very important information. What can be the next line of treatment for this very specific patient? To set up such a database, of course, it has a lot of interactions. So one of the given interaction is already with the existing molecular databases. So all the molecular genomic information of cancer patients are already stored in databases and are accessible for research. but all the other information has to be given in manually by different user groups. So for patient administration or for a single patient view, all these people have to enter data into such a database. And then, of course, you can imagine that reporting is possible on different levels, on the patient level, on the hospital level, on the statistical level, so we even can measure KPIs. I'm always saying today in our hospital, even at the University Hospital in Zurich, we do not know how many lung cancer patients are treated today in the hospital or on the current way in the hospital. I always joking and saying only the financial department knows because they are sending out these invoices. Yeah, but if you ask any oncologist how many lung cancer patients are treated today in the hospital, nobody can tell you. Yeah, so this is really the theme we are talking about, that all the information is there, mainly stored in PDFs, but nobody can really access and get good overviews about the information on a general view. So we were thinking about how to set up such a database, and obviously we came immediately to the HL7 FIRE language for coding this information. So this is a standard coding language for health topology or terminology, and everything can be coded. Every state of the patient, every drug, every treatment can be coded in a very free way. It's freely available, these languages, so you don't have to pay any license. And as you can see on the right side, that already many companies are using this kind of coding language to code health data. There's already out the M code, which stands for Minimal Common Oncology Data Elements. So here in the oncology field, we are maybe a little bit more advanced, so that already we have a system, a standardized system, how things can be coded. And with the different colors, you can see the different themes of coding. So for example, here is a disease described very well. The blue part is a treatment. And the green part is a genomic profiling, which is coded down here. And in the center of the whole M-code system stands a cancer patient. So that the patient, really everything comes together to the patient, and the goal of this project is in the end that the oncologist can decide on digital data how to treat the patient best and how to move forward with this one patient. This shows the M code in a little bit more sophisticated way. So it's hundreds of different fields which have to be given the information for the database. How to set up such a database in the environment of the hospital. This is another very critical topic, and this took us even longer than to code the software for the database, that every stakeholder of a hospital, of the IT departments, are involved into such an endeavor, so that in the end the database is really integrated into the hospital's landscape of different IT systems, so that such a database can grab the data from other databases as well. Now I want to come and I want to do a live session as well. That's why I have brought up my computer here to the stage. But first, 2 or 3 slides about how it looks like, so that you also can navigate with me together in a few seconds through the database. So this is a welcome page. This is you're personally addressed here, so that means it's a personal login, so that only those people who have the right to log in and to see the data can log in the database. Everything is tracked. That means if you logged in and you look for specific patients, this is tracked, and you can even ask later why you have looked up this specific patient. This is not your patient. What is the interest that you want to see the data of this patient? Then we have down here different fields. So I will go into the patient field where all the information is put in. We are now working on the data analysis, so really on the horizontal view that many patients and the data of many patients can put together for research projects and research analysis. And then, of course, and maybe you had some good ideas here I can take up, future features are always welcome. When we are going to the patient level, you see the patients and the disease of the patient. You see the birthday, which actually is already a critical topic. I mean, for Latvia probably as well as for Switzerland, you have to imagine how many people are born each day. It's maybe 10, 15, 20 in a 2 million population size. That means with a birthday already, you can track down a specific person in your country. You only have the choice of 10, 20 persons, that's it, and then you have already a very small cohort of people, and that means you can really track down very easily a birth date. So we are thinking about to even anonymize a birth date and just to give the birth year into such a database so that there is no tracking possible towards individual persons. And then you're coming to an overview page with all these fields, and I think we do now the live view so that I can show you how this looks like. So thanks to the technical people, we have managed this in the morning that it should work. All right, so here we are back to the database. As you can see, it's a landing page, and now we are going to the patient's view. And here I have now the choice to model patients, and we are going to this one for the lung adenocarcinoma. And if you move, and this is now really live, so I'm just entering into our database in the hospital via VPN, and can see all the information of this very specific patient. So they have the case overview here above, and then you have all these different fields, and you can open these fields, and you can see what happened in time-wise. So you have also a little bit a time schedule. But I think to get an overview, much more important, is really these fields down here, where all these information which are gathered even from different clinics in a hospital. I mean, a university hospital of 8,000 people, I don't know, we have 1,000 physicians. The patient is moving from one clinic to the other one, and typically an oncologist does not really know what happened in a radiology clinic, or what happens if it's prostate cancer in your urology clinic. So this is really hard to get these kind of information. Typically you call the physician in the other clinic, you ask for the PDF file, which is quite cumbersome. And with such a system, you're able to really oversee the information within one click. And then you can go into these fields and really look what is the state of the patient. And according to M-code, we have done the characteristics in several fields. So in the beginning, you see the cancer characterization, then the treatment information down here in the second field, and in the third one, the clinical events, what happens during the medication treatment, how successful was the treatment, and down below the pathology information with all the medical genomic information plus all the IHC and the other information the pathology typically reveals to a cancer patient. Yeah, this is now the start of such a system, and actually one big problem not only is really today, who is entering the data. The interface between man and machine is still probably the most critical and most cumbersome interface, because we have really hired medical documentaries who are starting to put in all this information from PDF files, because typically the oncologist says, no, I don't want to mess around with such a database. I'm much faster writing everything on a PDF, or even on a piece of paper, which is then later PDF'd by the administration. So we have hired these medical documentaries. They are entering these data, and the approval of such a data set is then done by the treating physician. So the treating physician receives an email which says there is new information for a specific patient available, please check. And if the physician approves this information, then the information goes into the database and is accessible for all physicians for further information. Yeah, thank you very much.
Thank you. Thank you. And we will welcome next speaker, Brian Haley Bird, Senior Lecturer, the School of Medicine, University College Cork, and Consultant Medical Oncologist in Bone Hospital, Cork, Ireland. Please.
Thank you. Oops, right. So, sorry, I'll go back. That's where I work, and I'm here to learn as much as I am to share my experience. And I'm fortunate to work with a great team of colleagues, pharmacists, and research nurses. And we're lucky also to have a new cancer centre. So these are my disclosures. And a bit of background on Ireland. We're also a small country, and we've got a fragmented healthcare system, and there is universal public healthcare, but there's also a lot of private insurance, and I work in a private hospital. And at the moment, the insurance companies will pay for EMA-approved drugs, sometimes many years before the state will pay for EMA-approved drugs. And newly, a lot of the insurance companies will cover a foundation medicine test with pre-approval once a year. So we have a very active and strong cancer society that has led to a lot of public health measures and better support for patients. And I really think that patient engagement is crucial to the success of cancer treatment in our country. So a bit of background on my practice. Because of socioeconomics, a lot of my lung cancer patients have never smoked and will have a druggable mutation at about twice the frequency seen in the public sector. My patients tend to be educated. They're pushing me for NGS, and sometimes they want to buy them even perhaps ahead of the data. And sadly, sometimes they buy drugs themselves, and that obviously raises ethical questions. And I'm very much supported by my pharmacy colleagues. So we had 6 patients on adagrasib, but the data is siloed, so I can't search the database and see which of my patients have G12C mutation. I just have to keep an eye out as somebody's lung or colon cancer progresses and go back and check manually, do they have such a target? We always like to treat patients on clinical trials to get data that everyone can learn from where possible. So when I started, when I finished up my fellowship in America, at a molecular level, things were relatively simple. There weren't that many targets, and even some of the stuff like RAS-RAF mutation in colorectal cancer was more prognostic than predictive. But this is the panel which my colleague Stephen Finn does in Dublin, in one of the big Dublin hospitals where we send our cancers for molecular testing, and you can see that things have got a lot more complicated. And so for lung cancer alone, we're looking for the Oncomine panel, PD-L1 tumor proportion score, and in patients who are progressing on a TKI, we will then do NGS. And this is an example that there can be a lot of difference between the genotype, this is my dog's genotype done by a commercial company, and the phenotype of the dog. So there are pitfalls when you order these studies. I hate clonal hematopoiesis because it really confuses me if I don't know whether the blood DNA test is coming from an aging mutated white cell or the tumor. There's hereditary cancer syndromes, and even if you order a somatic panel, you can pick up hereditary cancers, and that opens medical, legal, and ethical trouble. A lot of the time, sure, the gene is there, it's actionable in one cancer, but is it really actionable in your patient's cancer? We don't have enough DNA shed from the tumor into blood. What do you do with a very small signal? And then sometimes you know what the patient needs, but there's no way to access it. And I think it's disgraceful that I can't send patients in Cork to France or to the Netherlands for a clinical trial because the Irish government won't cover what are called non-trial costs. So the barriers to NGS are funding, availability of drugs if we find a target, and a genuine fear among me and my colleagues that we will misinterpret the data. And this is just a slide about clonal hematopoiesis. So you not only get tumor DNA shed into the bloodstream, but you get mutated white cell DNA that's a minor risk factor for developing leukemia or heart disease shed into the blood, which confuses the innocent oncologist. So how can I cope with this? Well, you know, like all of you, you build experience, you read the literature. When you get a new genetic result of a gene you've never heard of, you look it up on ClinVar. But I really recommend molecular tumor boards. So now our molecular tumor board is run through Cancer Trials Ireland, our cooperative group, and it empowers clinicians to make better decisions to help patients. So about 5 years ago in Munich, I had a discussion with Dee Peretti from Roche and Dervla Collins, my colleague in Cork, and Roche funded a pilot project, and ethically they paid Accenture to run it so that they would never hold even pseudo-anonymized patient data. And we have a panel of Irish and international experts, and we comment on 1 to 3, now 1 to 4 cases in an hour. It's now moved under the Cancer Trials Ireland umbrella, but it's supported by a variety of industry contribution. And recently we've moved to getting a pseudo-anonymized formal report that can go in the patient's file. And this is very helpful because when the patient progresses in 3 or 6 months' time, I have an accurate, unbiased report of what was documented, because you may think you'll never forget this discussion, but when you go back to the patient's chart, you'll find you certainly have. So this is pretty much the format where we enter the patient's clinical details, pseudo-anonymized, into a PowerPoint template, and it forces the clinician to think logically about the case and to outline previous treatments, and then we send it to the experts, hopefully 10 days ahead of time. And these are some of the experts: Rodrigo Dienstmann in Barcelona, Stephen Finn, a pathologist in Dublin, Marie-Dominique Galibert in Paris, and Terry McVeigh, who's a wonderful clinical oncologist. And Terry has set up an online education program for clinicians in the field of cancer genetics, which is run through the Royal College of Physicians in Ireland. So what have I learned from the MTB? I've learned when a new mutation is likely to be actionable, such as an EGFR resistance mutation. I've learned to fear and avoid CHIP. I've learned when, yeah, that's interesting, but so what, as in a PIK3CA mutation outside of breast cancer. More and more patients who are being referred, or their family are being referred for hereditary cancer genetics, and a new concept to me of orthogonal testing. And it can be very powerful to go back to immunohistochemistry if there's a genetic signal. So looking at a 1-year snapshot, most of the cases we discussed are lung cancer, after that GI and hepatobiliary, and then some odds and ends. So the MTB has changed over time. At the start, I think the questions I and other clinicians asked were fairly naive. Now we at least think we're more sophisticated. And so if you're an expert advising a molecular tumor board, you're going to have to acknowledge there will be a learning curve among clinicians. And now we're also getting feedback between clinicians over, yeah, when I use this drug, I saw this toxicity, or just because this patient had a toxicity with drug 1, they won't get the same toxicity with drug 2. So the feedback can be across centres who are dialed into the MTB. And industry also benefits both in terms of R&D, market access, and strengthening relationship with medical managers and clinicians. So this is a real MTB case, and when I presented this to my wife and daughter, who are my harshest critics, they said I had to summarize it because there was too much data. But it's a great lady who gave me her permission last week to present this case here, and I've been treating her now for 8 years with metastatic colon cancer. And once we treated her with panitumumab and her tumor progressed, she paid for a Guardant 360 test, and that showed elevated HER2 copy number. And that was her first discussion several years ago, and the MTB recommended that we do HER2 immunohistochemistry, which was strongly positive, and then we got compassionate access trastuzumab, pertuzumab from Roche, as it happens. And that didn't work, but she had good quality of life on it. And when she progressed, the Molecular Tumor Board recommended a very expensive drug, trastuzumab deruxtecan. And there was, for a long time, compassionate access for breast, but no other cancers, and even that access is now closed. So when she progressed again on chemotherapy, she cashed in her life insurance to pay for trastuzumab deruxtecan. And really, I wasn't pushing for this, but it was her choice, and she got 4.5 months with reasonable quality of life out of trastuzumab deruxtecan. So if anyone's got any radionucleotide labeled trastuzumab or anything interesting, she'd be interested. So this is the format of the case. When I present it to the MTB, we have to put up a summary of all previous lines of treatment so that we know what an appropriate recommendation might be. I think it helps to put up a case and to put up some imaging. So let's see if this works. You can see there's a mass in her bladder. That's what we biopsied on 2 separate occasions. And unfortunately, she also had a mass invading her sacrum, causing pain. And she's always had very radiosensitive disease. And that's responded nicely to radiotherapy. So the initial test was she didn't have MSI high. She had the HER2 amplification. And then that's her tumor immunohistochemistry, H&E. There's the HER2, strongly positive. And then this was her most recent foundation medicine test, which her insurance company paid for. And then the format of our MTB is that we're encouraged to ask a methodology question as well as a question tailored to the patient case. So I wanted to know, how can the tumor, which is still strongly expressing HER2, be ignoring trastuzumab deruxtecan, the drug antibody conjugate? What was the next line of treatment? And then there's meant to be a slightly better drug, that there may be a compassionate access window opening in Ireland, a dirty TKI, but it's better than nothing maybe. So Marie-Dominique talked about the various mechanisms of resistance, not just receptor downregulation, but also SLX4, which is a gene I'd never heard of, and the different ways in which different HER2-targeted therapies can be overcome by cancer evolution. And SLX4 loss may be a way in which my patient's cancer is escaping from this targeted therapy. But I had thought that the patient's MET fusion, which was hidden in the molecular result, was interesting but not actionable. But Rodrigo thinks that this non-canonical MET fusion may actually be actionable. And we're lucky through Stephen to link in to Theodora Mladic in Amsterdam. And she's agreed to do research MET immunohistochemistry testing and maybe even fusion testing. And perhaps if we do find this rare lung cancer target, we could give her off-label crizotinib. But her kidney function is very borderline. I may be able to give her low-dose crizotinib. And that's where, being a clinician and accompanying your patient on their journey and knowing about things like renal and hepatic function, still not quite yet replaced by AI. So this has been our MTB journey, from preliminary discussions to a pilot and now to a more formal pseudo-anonymized database. So we're going to hopefully use it to generate public funding for NGS in public patients, to formally allow off-label prescribing, refer to clinical trials, and prioritize referral to our underfunded hereditary cancer genetics service. These are my suggestions from what I've learned, and maybe you can avoid some of our errors. Do have a template, decide what cancers should be allowed in. If you've got enough of a population, like for example, Italy has a gynae-only molecular tumour board. Decide about the level of consent and data protection that are needed. It's great to get a printed report. It's also nice to get continuing medical education points. And I think we should get patient consent to prospectively gather data on an online database to get published. And also we're hoping to join the European Molecular Tumour Board network. And this is the European Molecular Tumour Board. And so perhaps all over Europe we can come together to drive precision medicine. So look, it'd be great to have a geneticist and a data scientist at every MDT. It would be great if we could sequence everything. Cork has got particular expertise in fecal microbiome, and then the AI would give us a hint of what to do, and maybe the hospital pharmacy will have a 3D printer that prints out the molecule the patient needs. But a bridge to that future is the molecular tumour board. And perhaps even though, as Dr. Gisselsson said, the percentage of patients who benefit is low, it is also reassuring to patients and their families that nothing was left undone in the quest to use modern medicine to help them. So this is the team, even radiation oncologists, they're very good, who I'm fortunate enough to work with. And thank you very much for listening. I'll take questions later. Thank you.
Thank you. Next speaker is from Latvia. Professor Aija is the head of the Cancer Biomarker Group at the Latvian Biomedical Research and Study Center and a professor at the University of Latvia in molecular genetics and immunology. Please.
Hello. I'm really very happy to be here today and to present our research on extracellular vesicles as a source of cancer-derived biomarkers in liquid biopsies. So I'll switch gears from the molecular tumor boards to the research on liquid biopsies. How do I? Oh, okay. Extracellular vesicles are membrane-bound vesicles that are released by a variety of cells, including cancer cells. There are three main types of extracellular vesicles. These are exosomes, microvesicles, and apoptotic bodies that all differ in their size, molecular composition, and functions in the body. EVs contain various lipids, proteins, RNAs, and even DNA fragments and various metabolites. And initially, they were found as a kind of waste disposal mechanism. But currently, it's clear that this is one of the main intercellular communication mechanisms that cells use to send various messages. Cancer-derived EVs have been shown to promote cancer development in a variety of ways. Cancer-derived EVs can be taken up by other cancer cells, and then they stimulate their proliferation, migration, drug resistance, stemness, and hypoxia tolerance, and so on. They can also induce various effects, paracrine effects in the tumor microenvironment and lead to the reprogramming of the tumor microenvironment. And they can also elicit a variety of systemic effects such as immunosuppression and establishment of pre-metastatic niche. And cancer-derived EVs are present in all body fluids that are in the contact with a given cancer, and they have been shown to carry various cancer-derived molecules. And this is the reason why cancer-derived EVs have attracted such a great attention of researchers as a source of biomarkers in liquid biopsies. In fact, they may have various advantages over conventional analytes in liquid biopsies, such as CTCs or cell-free DNA. So they are highly abundant in biofluids as compared with CTCs. They protect the cargo from the degradation, and they may contain molecular signatures of parental cells. However, the main challenge in using these extracellular vesicles is that biofluids contain a mixture of EVs that are produced by various different cell types. However, the fraction of cancer-derived EVs in blood plasma or urine is not really known and is likely to be variable among patients. My lab is focusing mostly on the RNA cargo of EVs in patients with prostate and breast cancer, and we have carried out a number of studies in longitudinal samples from these patients. In the case of prostate cancer, we were enrolling patients at the time of diagnosis, and we were collecting plasma and urine samples before surgery, 3 to 4 months after the surgery, and we also collected histologically verified tumor and normal prostate tissues. We isolated the EVs from these biofluids, and as it has been shown before that cancer patients may have an elevated levels of EVs in the blood, we expected that the concentration of EVs should go down after the surgery. But this is not exactly what we see both in plasma and urine, so basically there are no major differences when we compare the EV levels before and after the surgery. And we also did not observe any significant differences when we compared the EV levels in patients with prostate cancer and with benign prostate hyperplasia. So we did RNA sequencing of plasma and urinary EVs and tumor and adjacent normal tissues. And the way how we tried to identify cancer-derived RNAs in EVs was the following. We were looking for RNAs that were overexpressed in the tumor tissue as compared with the normal tissues, that were present at relatively high levels in the pre-operation EVs and decreased after the surgery. So we expected that if the RNA level decreased after the surgery, it should come either from prostate or from prostate cancer. And if it is overexpressed in the tumor tissue, it should, well, its main source should be the prostate cancer. And we were looking for such biomarkers across all RNA biotypes. The richest source of RNAs turned out to be mRNA fragments, and we could identify 63 mRNAs that matched these criteria. But there were a number of such RNAs among long non-coding RNAs, pVRNAs, and microRNAs too. So we selected around 20 of these biomarker candidates, and next we tested their biological, their diagnostic significance for discriminating prostate cancer from benign prostate hyperplasia. And the two best candidates came out to be two mRNA fragments. These were NKX and GLO1 that showed statistically significant difference in plasma EVs between prostate cancer patients and BPH in two independent cohorts. So when we combined these two biomarkers, they could discriminate prostate cancer from BPH with AUC around 0.85 in the test cohort. It was slightly lower in the validation cohort. It was 0.71. Nevertheless, it still is by far better than PSA in this cohort of patients. However, obviously, we are not really there yet. One strange thing that I forgot to mention is that all these biomarkers showed much better performance when analyzed in plasma EVs, not in urinary EVs, which was exactly the opposite that we saw in our RNA-seq data. So the RNA sequencing study demonstrated that urine is enriched with prostate or prostate cancer-derived EVs as compared to blood, and therefore urine could be a superior source for EV RNAs for the diagnosis and active surveillance of prostate cancer. But it is unlikely to be suitable for post-operative monitoring of prostate cancer progression. Nevertheless, in the PCR study, we obviously failed to find an optimal normalization technique for normalizing urinary PCR assays, and therefore plasma assays showed much better performance. Although the fraction of prostate cancer-derived EVs in plasma is lower than in urine, it's still sufficient to allow detection and monitoring of prostate cancer-derived RNAs. So in the breast cancer study, we enrolled 30 breast cancer patients undergoing neoadjuvant chemotherapy. And we collected blood samples at the time of diagnosis, then several blood samples during the neoadjuvant treatment and after the surgery. And in this case, on the contrary to the prostate cancer study, we saw that the EV levels in blood of these patients strongly correlate with the clinical events. So in patients at the time of diagnosis, the EV levels were significantly higher than in healthy controls. They increased during the neoadjuvant therapy. 7 days after the surgery, they are still high, but then they go down to the levels of healthy controls 6 and 12 months after the surgery. So the approach how we searched for cancer biomarkers in breast cancer cases were slightly different than before. We were looking for RNAs that were increased in breast cancer patients as compared with healthy controls, and that were present in the pre-operation samples and decreased after the surgery. So we identified 7 mRNAs, 23 SNO-RNAs, 33 microRNAs, and a number of biomarker candidates in the other RNA biotypes. So in all these cases, we can see that these markers are highly specific to breast cancer patients. They are almost zero in healthy controls, so they decrease in the post-operation samples. However, they are present only in a relatively small fraction of cancer patients. Hence, individually, these markers have very high specificity, but rather low sensitivity. Then obviously the next thing that we could try to do was to develop a biomarker model by combining 8 different biomarkers that could discriminate breast cancer from healthy controls with the AUC of 0.90, which is a pretty impressive value, and leave and out cross-validation showed more or less the same, which suggests that this model is rather robust. We're currently trying to transfer it to the PCR assays, but so we'll see what comes out from this. We also looked for RNAs that could predict response to the neoadjuvant chemotherapy. So we compared the RNA profiles in responders and non-responders to neoadjuvant chemotherapy at the different time points, at the time of diagnosis and during the neoadjuvant treatment. And we found 6 microRNAs that overlapped in these 2 time points that were increased in non-responders as compared with responders. And they were high in non-responders at the diagnosis. After the neoadjuvant treatment, they were still detectable in some cases 7 days after the surgery, but they decreased in most of the cases 6 and 12 months after the surgery, thus strongly suggesting that these microRNAs are functionally implicated in the response, or in this case, in the resistance to chemotherapy. So what we can conclude from the breast cancer study is that a substantial fraction of plasma EVs in breast cancer patients are produced due to the disease process or the treatment. I'm not saying exactly that these EVs are produced by cancer cells themselves, but they are there due to the presence of cancer and due to the treatment. And EV RNA cargo consists of various RNA biotypes, including SNO RNAs, snRNAs, PVRNAs, transfer RNAs, and so. And these all biotypes represent promising sources of biomarkers, which has not been explored so far extensively. And we showed that the RNA cargo in breast cancer patients is altered as compared to cancer-free healthy controls, and it dynamically reflects the clinical events. And we have identified a set of RNA biomarkers, however, they still require further validation. And at the very end, I just wanted to touch another aspect or use of extracellular vesicles. One thing that I didn't mention at the very beginning was that when we look at the RNA-seq data from extracellular vesicles, then only about 50 to 60% of these reads, in fact, can be mapped against the human genome. Then 40 or even more should come from somewhere else. One thing that we expected is that somewhere else, this could be some contaminants coming from the reagents, environment, personnel, and so on. And therefore, we included non-template controls started at various time points during the library construction in the sequencing study. And indeed, when we subtracted all these, all these negative control libraries indeed contained some RNA reads. And when we subtracted all these reads that were present in the controls and the reads that included these fragments, then in fact we removed something like 40 up to 50% of those unknown reads. Nevertheless, we still have something like 40 to 60% left, and these are several millions of reads, and we went ahead and tried to find out what are these extra human reads. So the first thing that we looked at is, I have to say that there are no techniques that really allow to identify these EV RNA reads because they are very short, and the standard tools for the identification of microbial reads doesn't work for them. Therefore, what we did is we first tried to identify or characterize the microbiome of the tumor tissues of the same patients. So we had the RNA-seq data from the tumor tissues, and from that we used a tracker tool to identify the microbial species that are there. And indeed, in prostate cancer tissues, we found 178 various microbial species, most of them bacteria, but there are some fungi and viruses too. And in breast cancer tissues, we found 66 species. We also sequenced normal tissues. We did not observe really major differences between prostate cancer and normal prostate tissues, or between breast cancer and normal breast tissues. However, the microbiomes in breast cancer tissue and prostate cancer tissues are completely different. There are almost difficult to find any common species. So then the next thing that we did, we built a metagenome from all those microorganisms that we found in the tumor tissues, and then we mapped the EV reads against these tumor tissue microbiomes. Almost all of these species that were found in the tumor tissue were represented in the EV RNA. And then when we started to analyze the EV reads that mapped against these microbial species, and we compared prostate cancer EVs against healthy controls, we could identify RNAs that represented various bacterial species that were differentially represented in patients and controls. And what was most striking, that various pseudomonas were much higher in cancer patients. When we compared pre-operation and post-operation samples, many pseudomonas that were present in the pre-operation samples decreased in the post-op samples, which suggests that these pseudomonas RNAs, that the presence of pseudomonas RNAs in prostate cancer plasma are due to the presence of cancer. And once the cancer is removed, their levels decrease. Similarly, also in breast cancer patients, when compared to healthy controls, there again were pseudomonas and some other species like Micrococcus and Acinetobacter that were increased in patients at the time of diagnosis as compared with healthy controls. And we also could identify one microorganism which was much higher in non-responders to chemotherapy than in responders, and we may speculate that the presence of this microorganism here is somehow related to the response to neoadjuvant chemotherapy. So, and what we conclude from this part is that a significant fraction of EV-RNAs are derived from the human microbiome. In total, in prostate and breast tissues, we identified 365 and 93 microbial species. All of them were represented in the EV-RNA. However, all these species were also present in the gut microbiome. So, and then we still don't know what is the major source of these RNAs in blood. Is this gut microbiome, tumor tissue microbiome, or other microbiome niches? And we also see that the microbial RNA composition in plasma EVs in cancer patients is altered as compared with healthy controls, and the abundance of pseudomonas RNA may have a diagnostic value both in prostate and breast cancer patients. So, and finally, I would like to thank the people in my lab who did the work, and our main collaborators from Riga East University Hospital, and thank you for your attention.
Thank you, and I would like to ask all lecturers come to the stage, and also representative from the patient side. We have 15 minutes for discussion. And I would like to ask auditorium if somebody have questions, please, that come to the microphone and start with your name, family name, and then the question. I think that we have one question. Yes, please.
Hello, thank you for a nice presentation. So I have a question for Aija, Line. I read some research a long time ago that tooth decay for women, like where is the canal involved, it's a cause breast cancer. Do you think it's somehow connected with this bacteria, what you found there?
Well, is it okay? Well, this is really a complicated question. I don't think there is a simple answer to that. Well, it's tempting to think that the presence of this bacteria may have a causal relationship with the development of breast cancer. On the same time, it may just reflect some shifts in that how the immune system. It could be possible that the cancer affects the immune system and may have some suppressive effect on the immune system. Therefore, some of the species may expand, and that's why this could be just a consequence. So.
No, and there was data that at first woman has very bad teeth, and then cancer comes later after a few years because of this chronic inflammation. And they found the same bacteria like in this tooth and breast.
I don't really think. Well, I'm not sure. I don't really see that there could be a direct link.
Hi, I think it's a fascinating area of research. I was going to ask Aija, has she looked at, pre and post neoadjuvant or pre and post immunotherapy EV profiles? And then I suppose, is there any correlation between the EV signature, the tumor microbiome, and the fecal microbiome? There's a lot of data to say that the fecal microbiome affects response to immunotherapy. But I think what you're discovering is really thought-provoking, but not quite in clinic yet.
Was this a question or a comment?
No, I wanted to know, have you looked at the pre and post signature of EVs before and after immune therapy?
Unfortunately not. No, we have not had access to patients undergoing immunotherapy.
I have another question on that. So what are your thoughts on causality versus correlation? Because there are some studies that depending on the tumor microbiome, you have a different response to immune checkpoint inhibitors. Because I'm in melanoma, so I have a specific interest in that. And we have seen that the tumor microbiome influences the response to checkpoint inhibitors. Have you thought anything that you could say because it could just be a correlation, as you said, that the tumor is kind of a better host for a certain type of species, right? So that would be more a correlation. But I'm obviously interested in, would there be a possible therapeutic angle one could go into? Or is that too far ahead?
I can only speculate on this. At the point we don't have any data on this yet, but this is something definitely we will be very interested to study in detail.
Okay, I would like to ask about real life and real patients, because our patients are interested to have this target treatment and very personalized treatment. And about those molecular tumor boards, we are starting in Latvia at the moment. It's the first steps in this. which patients we need to send to this molecular tumor board. And maybe we will think to do this much more, not only patients who are not respond to treatment. Because we have guidelines and first line, and everybody who treat patients knows the first line is very important. If you have this golden first line, sometimes everything is okay, and then you have relapses and 1 second and so. Maybe we will start in the beginning with this profiling of patients and have some risk patients who need this target treatment in the beginning, not with the guidelines, but like individual treatment.
Yeah, maybe I can answer that. So we have at the University Hospital around 40 to 50 tumor boards. So it's not only the molecular tumor board, it's really many different tumor boards. And actually that's one problem, and that again goes into what I have presented about the database, that the information sometimes does not come from one tumor board to the other tumor board. And sometimes we simply do not know what was the first line or second line treatment outcome of this patient, why he stopped treatment, for example. And then the oncologist has to go back and look into the data and find out what was the reason. I mean, it's there, but not always for a tumor board instantly obviously visible. The molecular tumor board we are conducting, we send only those patients who get comprehensive panel testing. So we do not look into the patient in the tumor board if there are just 1 or 2 genes like a DILA or something else was done to define a very specific mutation. But for a comprehensive panel test, because the richness of information is so big, we are always saying that one specific scientist or physician cannot cope anymore with the data. And here really you need the swarm intelligence to define what is the next line of treatment and what is a recommendation of such a tumor board. That's how we deal in our hospital. And I can tell you we have every week around 20 to 30 patients in such a tumor board.
So I think it depends whether you're resourced to do what Sweden is doing and to do it up front, or if you've got limited resources, you have to triage. And I mean, I really think the low-hanging fruit, which is where we find most of our actionable success stories, is lung cancer, particularly in non-smokers or light smokers. So, if you have to triage, you do lung cancer up front, and maybe you do other cancers on progression. But if you're set up well, then it would be great to follow the Swedish model.
Could I comment? Yeah, thank you for raising the Swedish model. I'm really happy. So actually, we also do some triaging, and we think that just like you said, Brian, is that the molecular tumor board has a certain educational value. So what we tend to select away, to triage away, are clear findings. Other could be that we find nothing that adds anything. Then we just report it out. So geneticists, they just write out the report. I never see it. It's just out there in the digital system. And conversely, if there is like a standard of care, somatic mutation, like an ALK mutation in neuroblastoma, we don't really have to have that at the board either. It's the gray zone cases where we have like rare findings that might or might not mandate the treatment, or that makes us discuss the diagnosis. Those are really the cases that we spend a lot of time on as well. Because that is largely actually because of the educational values, that next time we have such a patient, we might not spend time at the board discussing the case.
In this context, I have a question to David, because we are also doing starting in our hospital now whole genome sequencing for tumor patients. So we are now establishing the whole procedure and validate the testing, and I'm pretty sure in a few years we are doing whole genome sequencing on a routine basis. What I'm always suffering a little bit is when you compare the outcome or the information which you get from a comprehensive panel test to a whole genome sequencing. there's almost no delta in between. That means the patient is very, very well treated and diagnosed already with a comprehensive panel test. And for me, always a question I also ask in my hospital, give me any reason why I should do a whole genome sequencing in terms of what is additional benefit for the patient when I do a kind of expensive sequencing, because you have to pay much more than for a panel test. Maybe you have a good answer for me, and I will immediately go back into my lab and start working on it.
No, but it's first of all, it's different with childhood cancer because we have so many rare variants and it's so research intensive. So that's kind of the obvious thing. That what we do, so we actually have virtual panels. We can't look at all the genes. We would have a lot of variants of unknown significance. We have 1,000 genes in the somatic virtual panel and around 100 in the germline panel that we know are useful and actionable. And then, but we can, as soon as there are new research findings in the literature, we can just retweak our digital panels. So we don't have to make any new chemistry. So in that sense, the whole genome sequence is a kind of investment for the future. So we rig like a sustainable future system because we do expect prices to come down extensively. Now we have economies are strained and there are now 2 new wars going on. But it will, after a while, it will keep going down with things. So we think this is the looking towards the future where we can just re-rig the panels as the research environment changes and we get new game-changing mutations. We don't have to redo the chemistry. So we think that's one of the reasons that the government actually invested in this small pediatric cancer project, because they said for the future, when prices go down, this will be in the future also for adult oncology. And I think sarcomas is a field where this will come quite soon. Already a lot of large-scale RNA-seq is being done on sarcomas, which I think that will be the next field. Because, again, very rare diseases, lots of fusion genes.
Thank you.
Just maybe a question on that. I was quite surprised about you specifying that you had more cases than you thought where you had to revisit the original diagnosis. That is, of course, has direct impact on anything further downstream. So can you narrow that? Because I think the examples were both sarcoma. So is there something like, if you go into a certain area, you go, oh, you better do this sequencing because there's a higher risk. So are there gray zones or something you would pick?
Yes, you're spot on. There's soft tissue tumors. They are well known to be difficult to diagnose with regular pathology. So molecular tools have been there for decades. And this is just like a Rolls-Royce molecular tool. And also CNS tumors can be quite tricky, even though we now have DNA methylation that helps out quite a lot. But it still cannot really pinpoint all the druggable changes in CNS tumors. So I think those would be the two, But then in pediatric cancers, you also have really weird stuff that not even reference pathologists can say what it is.
We have spitzoid melanoma, which is always a pain. So, things like that. So it would be good if we had like a list where one could go, like there's a high risk or where it's difficult to diagnose on like histology alone. I think in order to improve outcomes. If the diagnosis is already wrong, you don't have a chance, basically, further downstream. So,
may I ask in between? Thank you.
Yeah. Hi. I'm Dimitris. I'm a molecular geneticist from the Children's Hospital, and we do quite broad profiling for the cancers, and sometimes we find rare somatic variants in the genes that are not targetable, but the mutation is novel, so it's not known as an oncogenic one. And so we have sometimes molecular tumor boards to decide whether we report it or not. And I'm curious how it's done in other countries, because as I understand, these future medicine, etc., tools just provide you a list of mutation, and then you have to deal on your own, what you do with them or not. So my question is basically, do you report those or not? And if you do, how do you deal with them later? Thanks.
Yeah, so we are reporting all targetable mutations, regardless of the variant allele frequency. I think this is really in the discussion in the tumor board, if you should go for a subset of specific clones. Here the question is tumor heterogeneity, to move forward with a specific treatment.
Yeah, I'm sorry, but the question was not the VAF, but the mutation that it's not proven as oncogenic, but like nearby in the same domain, but a novel one, not previously for gain of function, for example.
Okay, so we have the classification, and we, which was shown, I think, by you, David. And so we are reporting everything which is WUS and above. So WUS likely pathogenic and pathogenic. And the WUS are listed on an extra page below. And well, I always say if there is no other treatment options available for the patient, we start discussing the variants of unknown significance, which is highly speculative, and mainly there's not really something coming out. Sometimes even the WUS is classified as pathogenic. For example, if you have a BRCA mutation, which is not yet described in literature, which we find very often actually, it's a terminating mutation, then of course it's not a variant of unknown significance, even though it's up to now unknown, but we already describe it as pathogenic because we know it's a terminating mutation, and therefore it's definitely harming the HRD system.
Thanks. And it's manually corrected, or it's more of an automatic classification?
We're doing it automatically. So we have a pipeline, everything is done automatically, and then somebody is looking over it and manually curating the report and sending it out.
Okay, thanks.
Yeah, something that we have found very convenient as a tool. I mean, we work in a similar way, but case by case. But we also have a cohort of around 100 normal genomes that's been locally collected in the hospital registries, and they help us a lot actually to triage away variants of unknown significance. Because if we see them in one or two patients in the panel of normals, then we tended not to report them out. And that kind of helps us to get rid of some of the variants of unknown significance. Because of course, if you use databases, they are not from the south of Sweden, those genomes, so it's a difference.
Sorry, I just wanted to say that sometimes if your gut as a clinician says there's a mutation here and the panel comes back negative, it's worth repeating a biopsy, doing an orthogonal test, sending a new sample. So I recently had a patient who was a lifelong non-smoker with stage 4 lung cancer, female, not Japanese, but still, and her test came back negative. So we sent it out and it came back positive, and it turned out to be tumor heterogeneity in the same lymph node had gone to 2 different labs. And when this first lab retested the second sample, they straight away found the mutation. So sometimes as a clinician, you keep having to knock on the pathologist's door.
Yes, it's so. Yes. Okay, we have no time for discussion, and I would like to say thank you for talks and thank you for discussion, and we have a break at the moment. Thank you.